Welcome back, my dear students. Today we are going to journey inside the cell to uncover the amazing GERL system and endoplasmic reticulum, the cell's dynamic network for protein synthesis, protein modification, and protein transport. We will explore how the endoplasmic reticulum with its rough and smooth regions play a vital role in co-translational translocation, glycosylation, lipid synthesis, and chaperone-mediated protein folding. You will also discover how the GERL system linking the Golgi apparatus, endoplasmic reticulum, and lysosome ensures that proteins are correctly processed, packaged, and delivered to their destinations. By the end of this class, you will clearly understand how this interconnected system keeps the cell running smoothly, making it one of the most efficient biological assembly lines ever discovered. The image showing the endomembrane system, each organelle which has distinct structure and function within the cell membrane, like some organelles like endoplasmic reticulum, Golgi bodies, lysosome, and some vacuoles which are made up of membrane bound structure. They have function coordination among them, so they are basically called as endomembrane system. So they are actually GERL, Golgi body, endoplasmic reticulum, and lysosome. G for Golgi body, ER for endoplasmic reticulum, and L for lysosome. All of them are have membrane bound structures, and they have coordination in their functions. So this is the cellular structure. This is the nucleus. With the association with nucleus, there are endoplasmic reticulum, and there are some Golgi bodies, lysosomes, vesicles, etc. Peroxisome, mitochondria, and chloroplast. They are membrane-bound structure, but peroxisome, mitochondria, chloroplast, they are also membrane-bound structure, but their function is not coordinated. So they are not part of the endomembrane system. Endomembrane system consists of only Golgi bodies, lysosome, and some other vacuoles. So this GERL system is a coordinated complex of three organelles: Golgi apparatus, endoplasmic reticulum, and lysosome, G, ER, and L. The name GERL was introduced by Alex Benjamin Novikov in 1967. This structure is responsible for important cellular processes such as endocytosis, for intake of external materials, exocytosis, secretion of substances outside the cell, and third one is the synthesis, recycling, and waste removal. This is the cell plasma membrane. This is the Golgi apparatus, endoplasmic reticulum. There are some lysosomes, early endosome, late endosome, which ultimately form the lysosome. These are the cytosol or cytoplasmic structures, and from the Golgi apparatus, secretory vesicles move into the cell membrane. Now we introduce endoplasmic reticulum. Endoplasmic reticulum was discovered by K.R. Porter and Claude and Thomson in 1945. The term endoplasmic reticulum was given by Porter in 1953. Further electron microscopic studies was performed by George Palladay in 1974. Endoplasmic reticulum occurs in all eukaryotic cells except egg or ova, embryonic cells, and mature RBCs. But these endoplasmic reticulums are absent in all prokaryotic cells. They occupy about 10% of the total cell volume. During or isolation, endoplasmic reticulum fragments reseal into small vesicles called the microsomes. General structure of the endoplasmic reticulum. Under electron microscope, appears as an extensive membranous network spread throughout the cytoplasm. It forms a continuous system connecting the nuclear membrane at one end and the cell membrane at the other. The membrane increases the internal surface area of the cell 30 to 40 times. The or endoplasmic reticulum divides the cell into two distinct compartments: luminal space, which is located inside the extraluminal space, which is found inside the cytoplasm. And the internal space of endoplasmic reticulum is filled with fluid, which is called endoplasmic matrix. Morphological forms of or endoplasmic reticulum. Occurs in three distinct forms, three different structural forms: cisternae, tubules and vesicles. Cisternae are flattened, elongated, unbranched, sac-like structures. They are usually arranged parallel to each other. Many of them have ribosomes attached, especially in case of RER, ribosomal ER. They are found abundantly in cells with high metabolic activity, such as liver and pancreas. Tubules, they are irregularly branched, tube-like structures forming a network. Ribosome free surface are there in the on the tubules. They are common in cells engaged in lipid and steroid synthesis. Example: adrenal cortex, testes, and ovaries. Vesicles, they are spherical or ovoid structures. They are common in pancreatic cells and spermatocytes, where only vesicular endoplasmic reticulum is present. When cells are disrupted during centrifugation, air breaks into microsomes, which is called as also known as small vesicles. Types of endoplasmic reticulum: this is smooth endoplasmic reticulum where is no ribosomes on their surface, and these are the rough endoplasmic reticulums. There are ribosomes are found at the surface of the endoplasmic reticulum and which are responsible for protein synthesis. So smooth endoplasmic reticulum is also called agranular ER or agranular endoplasmic reticulum, where no ribosomes are found. They are abundant in liver, adrenal cortex, gonads, and muscle cells. Their surface appears smooth under electron microscope, and they predominantly made up of tubules and vesicles. Their main functions include synthesis of lipids and steroids like cholesterols and hormones, their carbohydrate metabolism, detoxification of drugs and harmful substances, especially in liver cells, pigment synthesis in certain cells, calcium storage in muscle cells as sarcoplasmic reticulum. Rough endoplasmic reticulum or RER, it is also called as granular ER due to the presence of ribosomes on its surface. They are mainly composed of cisternae. Few tubules are present. They are basophilic in nature due to ribosome content. They are prominent in secretory cells like pancreatic cells, plasma cells, etc. Their functions include protein synthesis with the help of ribosomes, processing and transport of proteins to Golgi apparatus, and glycoprotein formation via riboflavin 1 and 2 that anchor ribosomes. So what we found, that smooth endoplasmic reticulum is the portion of the endoplasmic reticulum where ribosomes are absent on the surface, and rough endoplasmic reticulum is also known as granular endoplasmic reticulum due to presence of ribosomes on its surface. They mainly composed of cisternae. Few tubules are present, but smooth endoplasmic reticulum is made up of only tubules and vesicles. The smooth endoplasmic reticulum uh, are found abundantly in liver, adrenal cortex, gonad, and muscle cells. But rough endoplasmic reticulum is basophilic in nature due to ribosome content. They are prominent in secretory cells. Example: pancreatic cells and plasma cells. Their functions include protein synthesis with the help of ribosomes, processing and transport of proteins to Golgi apparatus, and glycoprotein formation via riboflavin. Riboflavin 1 and riboflavin 2 are the two proteins that anchored the ribosomes on the surface of the endoplasmic reticulum. So what are the relationship between ACR and RER? They are interconvertible. ACR and RER, depending on the functional state of the cell, they can be converted into each other. Proportion about 2/3 RER and 1/3 ACR are there of total endoplasmic reticulum content. Stability: RER is more stable than ACR. Cell type determines dominance. Protein secreting cells have more RER, but lipid secreting cells have more ACR. So what are the modifications of endoplasmic reticulum? Annulate ER or annulate lamellae. McCulloch was discovered in 1952. These are basically the ER having pores like nuclear pores. They formed from blebbing nuclear envelope. They can be smooth or rough. Sarcoplasmic reticulum, a modified form of smooth endoplasmic reticulum in striated muscle cells, both in skeletal cell and cardiac muscle cell. They supply calcium 2 plus to muscle cytoplasm. They help in intracellular impulse transmission and contraction. Myeloid body: ACR is in pigmented epithelial cells of retina of frog. It helps in photoreception. Nasal granules or tigroid bodies, the bunches of RER present in the cyton or cell body of a neuron. Functions: Common functions of the endoplasmic reticulum. Endoplasmic reticulum provides mechanical support to the cytoplasmic matrix and forms the skeletal framework. It provides a large surface area for the synthesis of biomolecules as they different enzymes. Here, or endoplasmic reticulum maintains the cell organelles in their proper position. Endoplasmic reticulum facilitates quick intracellular transport, thus forming the circulatory system of cell. Ear extends through plasmodesmata as desmotubule and control the movement of biomolecules between adjacent cells. So formation of nuclear membrane during cell division. So the protein processing and secretion is one of the main function of ear. Ribosomes assemble proteins from polypeptides entering through ear. Then protein move through the rough endoplasmic reticulum where they are further modified. Then transport vesicles containing the proteins are pinched off from the rough ear. Then transport vesicles fuse with the membrane of the Golgi complex and the proteins are released to the inside. Within the Golgi complex, the proteins are further processed and stored. Vesicles containing the finished proteins are pinched off from the Golgi complex. Then vesicles travel to the cell membrane, fuse with the cell membrane and release the proteins to the outside of the cell. Functions of RER. RER contains ribophorins for attachment of the ribosomes on their surface. RER synthesizes proteins destined for secretions and also involved in folding and transport of proteins. RER forms nuclear envelope, cell membrane and ACR. It takes part in glycosylation by the enzyme glycosyltransferases and disulfide bond formation within the protein molecules. Functions of ACR. ACR takes part in protein synthesis of lipids, glycogen, steroid hormones like cholesterol, progesterone, testosterone, etc., ascorbic acid. ACR helps in detoxification of toxins using cytochrome P450. There are enzymes that modify toxic chemicals or carcinogens to reduce its toxicity are present in ACR. In liver cells, ACR possesses enzymes called glycosomes for glycogen metabolism like glycogenesis or glycogenolysis. Within the retinal cells, ACR produces visual pigments from vitamin A. In muscle cells, ACR modified into sarcoplasmic reticulum which store and release calcium 2 plus for muscle contraction. ACR is also responsible for the formation of cell organelles like Golgi apparatus, lysosomes, spherosomes, vacuoles, etc. These are the image showing the rough endoplasmic reticulum which contains ribosome and the smooth endoplasmic reticulum without the presence of ribosome. Protein processing in the endoplasmic reticulum which have three basic steps. First one is the co-translational translocation of proteins through ER membrane. What are these? The definition? Co-translational translocation refers to the simultaneous translation of a protein and its translocation into the lumen of the endoplasmic reticulum. What are the key steps? Signal sequence recognition. Ribosome synthesizes a single peptide at the end terminus of the protein destined for the endoplasmic reticulum. And the signal recognition particle or SRP binds to the signal peptide and halts further translation temporarily. Docking to ER membrane, the SRP signal recognition particle ribosome complex binds to the SRP receptors on the ER membrane. Protein translocation, the ribosomes found on the surface of the ER aligns with the translocon or Sec61 complex on the ER membrane when SRP is released and translation is resumes with a growing polypeptide chain passing through the translocon into the ER lumen. Cleavage of signal peptidase and enzyme cleaves the signal peptide once the protein enters into the ER lumen. What is the diagrammatic summary of the concept? Co-translational translocation occurs when ribosome releases the signal peptide which bounds with the SRP, then it moves into the translocon and into the ER lumen. This is the diagram showing the process of protein processing with the involvement of ER. Second point is the glycosylation of protein. Glycosylation is the enzymatic attachment of the carbohydrate group or glycosyl moieties to proteins, which is critical for proper folding, stability, and function. Types. Glycosylation can be of two types: N-linked glycosylation, which occurs at asparagine residue within the consensus sequence, This is the sequence asparagine, X serine or serine. It involves the transfer of a pre-assembled oligosaccharide from a dolichal lipid donor to the nascent protein. Olic glycosylation, it occurs at serine or threonine residues, which is more common in the Golgi than the ER. Process in the ER, glycosyl transferases enzyme catalyzes the addition of sugars, ensures quality control of protein folding. What is the diagrammatic summary? Glycosylation involves the dolichal oligosaccharide which transfer to protein and end-linked glycan. Third one is the chaperone-mediated folding of proteins. Role of chaperones. Chaperones basically assist in or help in achieving the correct 3D conformation of different proteins. It prevents aggregation of misfolded proteins. Key chaperones in the endoplasmic reticulum. First one is the BIP or binding immunoglobulin protein. They binds to hydrophobic regions of the nascent polypeptides to prevent premature folding or aggregation. Second chaperones in the ER are the calnexin or calreticulin. They ensure proper folding of glycoproteins by interacting with N-linked oligosaccharides containing terminal glucose residues. Protein disulfide isomerase or PDI, they catalyzes the formation and rearrangement of disulfide bonds. The fate of the misfolded proteins, which they are retained in the endoplasmic reticulum and targeted for degradation via ER associated degradation pathway or ERAD pathway. So the process ensures the synthesis of functional, properly folded and modified proteins within the ER. The chaperone-mediated folding involves the involvement of either BIP or calnexin or calreticulin into the folded protein. So my dear students, today we have discovered how the endoplasmic reticulum or ER serves as the cell's major site for protein and lipid synthesis and how its smooth and rough surface perform distinct yet coordinated roles. We learned how co-translational translocation, glycosylation and chaperone-mediated protein folding ensure proper protein formation and how the GERL system linking the Golgi apparatus, endoplasmic reticulum and lysosome acts as integrated network for protein modification, packaging and transport. Together they maintain the cell's internal organization and efficiency, truly a biological factory at work. If you enjoyed exploring how our cells manage such precise and complex processes, don't forget to like, share and subscribe for more insightful lessons in cell biology and life sciences. See you in the next class, and until then, stay curious and keep exploring the microscopic wonders of life. Thank you.